This is part 4 of the Molecules of Life lessons. Today's is about nucleic acids. Now, the last category of organic compounds that living things have is nucleic acids. Nucleic acids, as you can tell by the name, are acidic and they are found in the nucleus. There are two main types of nucleic acids. There's DNA, which is deoxyribonucleic acid, and RNA, which is ribonucleic acid. DNA is what makes up the genes on the chromosomes that are found in your cells and all other living things. The genes are instructions that code for that primary sequence of amino acids in all the proteins in every organism. The RNA takes those instructions that are coded in the DNA and uses those instructions to make the proteins. These processes are called transcription and translation. We will talk more about them later on when we spend more time talking about just nucleic acids and how they work. For the meantime, this is just an overview about them as one of the types of organic molecules. Now, the flow of information in the building of a protein goes from the gene, which is in the DNA, via transcription to RNA, to translation, which makes the protein. And again, we'll spend some time later on learning about how this works and learning more specifics about the particular process. But this just gives you an idea that the genes have the instructions are copied, transcribed into RNA, and the RNA instructions are used to actually build the protein molecules that make up your cells and every other living thing's cells. Nucleic acids are made of monomers, just like the other organic molecules we've talked about. The monomers in nucleic acids are called nucleotides. Nucleotides have three main parts. They have a 5-carbon sugar, a phosphate group, and a nitrogenous base. In DNA or deoxyribonucleic acid, the sugar is called deoxyribose, and in RNA, ribonucleic acid, the sugar is called ribose. Remember, OSE stands for sugar. The nitrogen bases, there are 4 different nitrogen bases in DNA and 4 different ones in RNA. 3 of them are the same. DNA has adenine, thymine, guanine, and cytosine. You can tell by the name that ene part means that nene or mean part means amino. So they'd have some nitrogen in there from an amino group. In RNA, we have adenine, uracil instead of thymine, guanine, and cytosine. The way they're put together is the sugar of one nucleotide combines to the phosphate group of the next nucleotide through the process of dehydration synthesis, as we've discussed with all the other organic molecules we've talked about. This makes a backbone for the molecule that is alternating sugars and phosphates. Here's what a nucleotide looks like, generally speaking. Here's our 5-carbon sugar, a nitrogenous base. Notice those amino groups on there. Okay, this particular one is adenine, and the phosphate group attached to the sugar. Now, when you put together a series of nucleotides, that forms something called a polynucleotide. And polynucleotides, like I said earlier, have an alternating sugar and phosphate backbone. So here we have the sugar, which is blue, the phosphate, which is tan, sugar, phosphate, sugar, phosphate, sugar, phosphate, all the way down. The nitrogen bases are attached to the sugars, but they're not part of the backbone. RNA is a single strand of polynucleotides, and DNA is a double strand of polynucleotides. In DNA and RNA, you have 2 kinds of nitrogenous bases. There are purines and pyrimidines. In DNA, the purines are adenine and guanine, and you can see here from the structure, they're both made of 2 fused rings with some groups attached to them. Okay, and the pyrimidines are thymine and cytosine. Thymine and cytosine, the pyrimidines, have only 1 ring structure. So the purines have 2, the pyrimidines have 1. When you put them together in a polynucleotide, those bases hydrogen bond together in the middle to form complementary base pairs. Notice this complementary with an E. That means they complete each other, not they tell each other how good they look today. Adenine always pairs with thymine, and cytosine and guanine always pair together as well. They do hydrogen bond in the middle, and as we said, and we'll see more about this when we talk more about DNA structure. Now, our RNA also has purines and pyrimidines, and the purines are still adenine and guanine, but the pyrimidines in RNA are thymine and cytosine. Are uracil and cytosine, I'm sorry. And so they would also pair up the same way if they were able to make a double structure, which they do and sometimes, and they certainly do when they are copying the sequence from DNA. But we'll learn a lot more about that later on. Now, when you look at DNA, you're used to seeing this double helix structure. And what you see is that pairing of the bases between the adenine and thymine and the cytosine and guanine here. And that's important for making copies of the DNA when the cell is getting ready to divide. These two strands of complementary DNA are held together by hydrogen bonds, the same kind of hydrogen bonds we learned about when we learned about water, between the positively charged oxygen end of one molecule and the negatively charged oxygen end of one molecule and the positively charged hydrogen end of another molecule. And that causes those base pairs to pair up just like that. They're attracted to each other by those bonds, and then they end up twisting around each other in that double helix because of other kinds of interactions. And again, we'll talk a lot more about DNA later. In this unit, we're just talking about it as one of these molecules, one of these organic molecules that make up the molecules of life.